Okay, so welcome back. I hope you had a nice lunch. So now we'll have a talk by Maggie Grundler from Berkeley.
Hi everyone, thank you so much for coming to my talk today. I'm really excited to present some results on the research I've been doing on dietary specialization and genomics in Thamnophis garter snakes. And dietary ecology is one of my favorite things to talk about, not only because it's just really cool, but also, in my opinion, it's one of the most important components of an organism's natural history. Not only does it provide the fuel for every other behavioral and physiological process that organism is going to undergo, it also kickstarts exposure to myriad selective pressures from competition and foraging. And so, of course, we see these many wondrous forms in our distant relatives across mammals, birds, insects, fish, including but not limited to all of these taxa, and my favorite, of course, snakes. But not only do we see striking interspecific diversity due to feeding morphologies or dietary diversity in general, but we also see quite a striking prevalence of intraspecific variation in diet as well. For example, these Placida dendritica sea slugs will starve to death if they don't get their preferred algae, even though conspecifics will eat the other type. Ruddy turnstones demonstrate multi-year consistency in one of 6 foraging strategies. And northern pike also demonstrates sympatric differentiation in foraging for fish and invertebrates. Not only is this super fun and cool, but it is also pretty evolutionarily significant. It can lead to increased network degree of food webs, which influences temporal stability of communities. This can generate a portfolio effect of trait diversity, which will stabilize populations over the long term. And if this variation is heritable, it can even lead to rapid evolution. But what does it really mean to be a generalist or a specialist? We kind of see in a lot of taxa this huge spectrum, right? And how and when and to what extent do species and populations and individuals really specialize? And you can think about, first of all, the fundamental niche, which might be influenced by evolutionary history. This can govern what an organism is able to detect, to capture, to metabolize. But if a population is experiencing environmental perturbations or competition, they might only be foraging on a subset of those resources, and this we know as the realized niche. But more than that, as I've just said, some individuals will demonstrate a preference. But why? And let's think about this first in a more quantitative context. So you can imagine this as a population distribution along a resource diversity axis, and there are several different ways that this can be subdivided by individuals in the population. You might have individuals that are broadly generalist, so they're eating many different things, and in resource diversity space, the distance between individuals is quite small. They're not really dispersed. Alternatively, you might have individuals that only eat a few things, and this leads to the opposite pattern where between individual variation is quite large, but within individual variation is quite small. And it's this question in general that particularly fascinates me. Why are some individuals in some species choosy? What governs that? Clearly there's some influence of evolutionary history. Underlying genetic architecture must contribute something, but environmental variability must also play a strong role. And so what I'm really interested in is teasing apart those mechanisms. And we really have a limited understanding of the molecular foundation of dietary ecology, but one of my favorite studies on frugivorous bats found that generalist foragers had a higher diversity of olfactory receptors. But I'm not a mammologist, so I want to study this in snakes, and luckily Thamnophis garter snakes also display a huge diversity of dietary ecologies and different modes of specialization within that. But my favorite are these snakes, this subspecies of Thamnophis elegans that specializes on banana slugs. So you can see here in the pink the range of Thamnophis elegans, or the wandering terrestrial garter snake. Inland, it's just elegans elegans, a broadly generalist species, but on the coast, where it overlaps with the banana slug distribution, you get this local sort of ecotype known as elegans terrestris. And they also overlap with another congener, Thamnophis ordinoides, which is a full slug specialist across its range. And so what I wanted to take a look at was how these differences in dietary ecology might be varying across this north-south transect of prey availability, but also this east-west divide of the two subspecies, and also what are the dynamics happening in that northern zone of sympatry, where you have this ecotype of a slug specialist with a fully more obligate specialist congener. So these are the species I'll be focusing on, and I'm going to use this color convention throughout the talk to refer to them. Thamnophis ordinoides is going to be in gold, and then I break Thamnophis elegans into three sort of different populations. There's the inland generalist population in red, the coastal specialist population that's sympatric with ordinoides in dark purple, and in violet, terrestris that is allopatric to ordinoides, but still overlapping with that range of banana slug prey. And I did recover stomach contents that confirm that we have this sort of spectrum of specialization where ordinoides is really only eating soft-bodied prey, elegans is eating a broad diversity of prey, and terrestris is eating banana slugs, which I didn't have an icon for, unfortunately. But they're also taking other kinds of prey as well. And so this is very interesting that we have kind of this, to what extent do we think they're specialists? How do we define that? And so I wanted to break this down, of course, into more detail, looking at what's going on across the seasons, and also comparing this to some metrics of genetic diversity. So for my main questions, I ask, how do these niche dynamics of these specialists compare? What genomic features are associated with slug specialization, and does this specialization contribute somehow to population differentiation? And briefly, my methods for this, for looking at niche dynamics, was to take samples for stable isotope analysis. Organisms incorporate the isotopes that are in their prey, and they, depending on the tissue, they metabolize it at different rates. So by taking two tissues and calculating the difference between those signatures, you get a proxy for individual specialization. And also, of course, you can calculate between individual specialization. To look at genomic features associated with this dietary phenotype, I looked at diversity across olfactory receptors that I pulled out of whole genomes that I sequenced for around 62 individuals. And to look at any contribution to population differentiation, I'm looking at genome-wide diversity statistics between the generalist and specialist ecotypes of Thamnophis elegans. So first I'll go through the results from dietary niche dynamics. On this graph, you're going to see on the y-axis a measure of between individual variation. And remember that the larger the between individual variation, the more dispersed individuals are in niche space. So it can be suggestive of individuals preferring different prey items. And in green and yellow are going to be early and late season sampling. Unfortunately, I don't have late season sampling for the inland generalist, but just to get you oriented, here they are on the axis, kind of somewhere in the middle, potentially suggested that they are slightly dispersed in niche space. Not necessarily everybody eating all of the things, but some preferring a few items, some preferring other items. For our full slug specialist Thamnophis ordinoides, they have relatively low between individual variation, which makes sense. They're eating soft-bodied prey. There's only so much they can eat. But this does reduce a little bit in the late season, so there is some variation happening there. With terrestris that's sympatric with ordinoides, you see this striking shift between seasons in dispersion and niche space. So in the early season, they're kind of mostly generalizing, and then in the late season, they're dispersing in niche space, and individuals have the opportunity to perhaps obtain preferred prey items, it's very interesting looking at that difference in dynamics between these two sympatric slug specialists. And in allopatric terrestris, we actually see kind of a reverse pattern, but not too big of a change between the seasons. What's important to note here is that between individual variation for the allopatric slug specialists is generally larger, at least in the early season, larger than the sympatric specialists. So they kind of have this opportunity to obtain preferred prey without being in the presence of that competitor, potentially. Looking at within individual variation can confirm some of these patterns. Again, I don't have full sampling for all of the subpopulations, but we do see this trend that within individual variation is decreasing in the late season for the slug specialist sympatric with organoides, and you do see much lower within individual variation for the allopatric terrestris. So the key takeaways from this part of the project are that, yes, our coastal subspecies demonstrates some preference potentially for banana slugs, but they also have this ability to really shift the dietary niche as needed. And for olfactory receptor diversity, I am just going to go over this kind of briefly because I don't have full genomic results, but for these preliminary results, we do see kind of this pattern of slightly higher heterozygosity across olfactory receptors for all of the ecotypes that are eating slugs. And if you look at this across the largest chromosome in the organism, the pattern is also holds true. So potentially there is higher standing genetic diversity in these slug-eating ecotypes. And for looking at population differentiation, I'm just going to focus on these two comparisons right here. This is the subspecies of elegans. Again, this is only the largest chromosome, and of course, very preliminary, it needs to be really dug into, but there's almost maybe a preliminary pattern of actually less differentiation between elegans and the northern sympatric terrestris slug specialist, despite the fact that, I got that backwards, less differentiation between inland elegans and terrestris south of that zone of sympatry. So despite this big kind of ecological barrier of the Central Valley of California, they're potentially actually more differentiated from the northern subspecies, which could indicate, I'll get into that later. So the significance of these results, we found that there's this kind of novel trophic shift without any obvious trade-offs of generalist predation. So Thamnophis elegans is able to kind of maybe shift between these different modalities. And this is really interesting to me because you see other patterns in nature of this evolution of really dramatic specialized structures with mostly generalist foragers. This smashing mantis shrimp has this amazing adaptation for crushing hard-bodied prey, but 30% of its diet is actually soft-bodied prey. So I think it's a really interesting eco-evolutionary puzzle. And potentially because of those very preliminary genetic diversity results, I'm sort of wondering if elegans might be experiencing local adaptation without evolution of reproductive isolation. And this can contribute to range expansion, perhaps, and population persistence. Thank you.
Hi, so I'm Carys from the University of Oxford. I'm doing a PhD there, and I'm going to talk about one chapter that I've been working on where I'm interested in thinking about how we can account for environmental similarity when we're estimating heritability. Like everyone in this symposium, I'm interested in exploring intraspecific variation and causes of that. And we see intraspecific variation across many different traits. It's important because it leads to variation in vital rates and varying life histories, which is important for evolutionary change and population resilience. So in order to explore the causes of intraspecific variation, one of the best ways to do that is having like in the wild at least, a long-term study system where we can monitor individuals over time and over space. And so I'm lucky enough to work in Oxford in Wytham Woods, which is this woodland in the UK, where we've monitored population of greaters, which are small songbirds, for over 70 years, where we collect every year various life history data. So on the right, there's a map, and each white dot is a nest box, and every year a number of field workers will go around and monitor each box and each breeding attempt. So over the years, we've got over 10,000 breeding attempts, and we can also identify individuals, so we can create this pedigree where we have over 35 generations of individuals. In particular, a trait that's super interesting is laying date in birds, so that's the date that they lay the first egg. This is, for these birds, a key seasonal timing trait, so they only nest once each year in the spring, and they need to make sure to time their laying date so that when the chicks are most hungry, there is most food available for them. So it therefore has like important fitness implications for the birds, and it's also advancing over time like many other phenological traits, and it shows variation between and within individuals. So this is sort of to illustrate some of the variation that we see over time and within years. So starting from 1960, each one is a year and just the distribution of laying dates within the population. The black line illustrates the mean over all years, and you can see on average the laying date is getting earlier, but some years it's still a bit later. And then there's also variation in the peak. So some have a really synchronous laying like 1987, and some like 2016, there's almost a bimodal distribution. So we see lots of variation. Also across space, so if you recognize the shape, each dot is just a nest box across the woods, and the color represents the mean laying date of that box over all years. And so you can see there's some areas where laying is often later and some where it's often earlier. And even if you zoom in to like a few little areas, boxes close together are often quite similar, but can also be different to each other. So yeah, if we think about what might cause this variation, we have our little greater in her nest box, and she's deciding when to lay her eggs. So it could be things like genetics that are influencing this, or many different sort of extrinsic factors, so maybe the temperature, or physical features of the environment, so the altitude, quality of her habitat, or like biotic factors, so the influence of the parents or a partner, population or breeding density around them, or like cooperation or competition with neighbours. So all these factors are influencing when this bird's deciding to lay, and separating out these genetic and environmental drivers can often be quite complex. And one way to start thinking about that is to estimate the heritability. So a super simple way to think about heritability is here you have this plot of just the similarity of related individuals. So along the x-axis is just a mean of like all mothers' laying dates, and then their daughters' laying dates along the y-axis. And you can use this relationship here to estimate, or the slope to estimate a heritability. But this then doesn't really account for any environmental effects that these individuals might be experiencing that may bias these estimates. So, for example, in our population, we know we have dispersal, or the birds disperse, so that's from how far they move from where they're born to where they then decide to lay their eggs when they're breeding. And overall, it has a median of like just over 600 meters and is mostly below a kilometer. So very often relatives will end up sharing environments with each other. And if we don't account for this, we might then assign some variation to genetics when it's actually just because these two individuals are sharing a similar environment and taking cues from that. So some more like complex methods to use that we can like estimate heritability are these quantitative genetic animal models. And we can use these to quantify the relative contributions of genetic and environmental factors. And what's really cool is we can take advantage of the huge data set that we are lucky enough to have, where we have this huge pedigree of thousands of individuals and how they're related to each other. And we can use this to create a matrix of relatedness between all individuals. And we can account for, instead of just like mother-daughter relationships, we can account for all different sorts of relationships between individuals. So we have our pedigree. We also can then account for things like fixed effects. So in this population, we know that older birds often lay, on average, earlier than younger birds. And yeah, so we're trying to explain this phenotypic variation. So this is a simple model. We also want to account for the random effect of breeding year. Like you saw earlier, there's a lot of variation between years, and there's advancing over time. What we're interested in estimating is the additive genetic variance of a focal individual. So this we can use to estimate the heritability. We also want to account for permanent environment effects of individuals. So this accounts for like repeated measures of individuals over time. And we can also estimate a residual variance. So yeah, what's the thing that I'm most interested in is then how can we account for these common environments shared by related individuals when we're estimating heritability. So one way to do this is we can include, on top of a matrix of relatedness of individuals, we can include another S or spatial matrix where we can account for similarities between the local environment of individuals. So I've thought about doing this in two ways. One's a most simple way, which is spatially, and another is thinking more about the breeding environment. So I'll go into these in more detail. So spatial proximity matrix, this is just the distance between all individuals, and overall this is assuming that individuals that are closer together will be sharing a more similar environment and therefore be more likely to lay at the same time, which is a very common assumption and is often true. But what if we want to consider more than just distance when we're thinking about this? So, for example, so I want a measure of environmental similarity. So, for example, if you think in this specific example, you have a woodland, and some birds, maybe there's a big hill, or two big hills, and you might have a bird nesting on the top of each hill in a very similar environment, and they're actually quite far away, but the environment they're experiencing is very similar compared to maybe a bird nesting at the bottom of the hill that by distance is closer, but the environment is quite different. So I think it's important that we think about how we can consider this on top of just spatial distance. So for our birds in particular, we know there's a few factors of the environment that influence when they're deciding to lay. So things like altitude or the slope that the box is on, the breeding density, proximity to the edge of the woodlands, and the number of oak trees. And we use some methods suggested by Thompson et al., where we can sort of combine all these measures that we know are important into one measure of similarity of environment between all pairs of individuals. So similar to thinking about a distance in space, we're just thinking about a distance in environmental space. Using this Euclidean distances. And overall, this is obviously quite related to spatial, but not super correlated with the spatial proximity matrix. So overall I looked at 4 different models. So I started just a super simple model where I don't account for any common environment effects. Then I start super simply by just including the nest box as a random effect. So this can account for, we assume that birds nesting in the same nest box over years will be experiencing a more similar environment to birds nesting in different nest boxes. And then these 2 last models where I include these extra matrices, so the spatial proximity matrix and the environmental similarity matrix. So a quick look at some results. So the colours should hopefully match to the side, and this is just representing the proportion split of variance. So when we have the most simple model here, we see that breeding year explains quite a lot of variation, which makes sense because we saw there was a lot of variation over years. And the blue on the bottom is the additive genetic variance, which we use to estimate heritability. And there's also a similar effect of the permanent environment, so repeatability of individuals. When we account for a nest box random effect, that's the little orange bar there, and we can see it explains a little bit of the variation we see, but not actually too much. It takes a bit away from the permanent environment effect, but not very much. But when we then include the matrix, the spatial similarity matrix, we see it explains almost 20% of the variation that we see in laying date, and reduces quite a bit the permanent environment and a little bit the additive genetic effect in blue. And when we include the breeding environment similarity matrix, it explains a similar amount as does the spatial matrix. So when we then go on to estimate heritability, so this is a within-year heritability estimate. The first 2 more simple models, you can see the estimate is around 20% with quite a bit of error, but when you include the matrices of distance and environment, it reduces the heritability estimate by quite a bit, by almost half. So overall, yeah, I just wanted to show that not considering these environmental effects might be quite damaging, and it might bias our estimates of heritability, which can then lead to other consequences when we're estimating evolutionary potential. And just to highlight the importance of thinking about ways that we can account for the similarity environments on top of just thinking about spatial distance and like accounting for the similarity environment experienced by individuals that aren't close in space. Next steps, not necessarily for me because I want to finish my PhD soon, but overall there was some really interesting stuff yesterday in the symposium about like the environment as an extended phenotype, and that's the sort of thing that in this context is quite interesting, like specifically thinking about if the environment in a way is also heritable. So for example, if individuals are inheriting territories or the habitat choice is something that's inherited, that could then mean by accounting too much for the environment, we're then like underestimating heritability. So yeah, there's lots of stuff there that could still be explored. But yeah, thanks so much for listening. Thanks to my supervisors and Whitem. And this is available as a preprint currently, if you want to have a look.
Really interesting talk. Thank you for sharing it. Have you considered including any interaction terms in your model? Thinking about, like, say, the interaction between spatial variation and genetic variation.
Yeah, that would be really cool. I haven't done it yet. One thing that's quite difficult, and like I don't want to sound boastful, but we have like so much data, so it's quite difficult sometimes to run some of the models when you have these huge matrices.
But you
So
have the power to enable more terms.
yeah, but yeah, so exactly, that is definitely something that could be interesting but I haven't yet.
Hello, thank you for coming for this talk. I am PhD student, finishing my second year, and today I'm going to present you some results of my work about plant defense suppression in spider mites. So we know that in nature, herbivores interact with plants to get energy and resources for their growth and reproduction. And under this pressure, plants evolve with a broad set of mechanical and chemical defenses, which consist in toxic or antidigestive or repellent compounds that reduce the performance of herbivores. However, in this complex interplay, herbivores also evolved a broad set of traits to overcome plant defenses. And these are generally divided into three categories: behavioral adaptations, metabolic resistance, or suppression of plant defenses. However, the origin and diversity of these offensive strategies are poorly understood, and surprisingly, there is lack of studies and evidence that these strategies are costly for herbivores. So to dig into this question, we focused on suppression of plant defenses. This consists in production of specialized molecules, also called effectors, which block the induction of plant defenses, bringing them down to or below the levels of uninfested plants. However, this was for the first time documented in plant pathogens, but it's also present in some arthropod species. But the suppression of plant defenses is not widespread among herbivores, which contrasts with the clear benefits that this trait confers to herbivores. And this raises the question, are there costs associated to plant defense suppression? And we may consider the physiological costs if the energy and resources allocated for the suppression compromise some life-interest traits as growth or reproduction. But this is not the only cost possible given the not straightforward outcome of this strategy. So on this graph, you can see the results of an experiment where authors measured the fecundity of two species of spider mites. On white, we have T. evans which suppress plant defenses, and in gray, we have T. urtica as a non-suppressor population. And the fecundity was measured on plants that were previously infested by suppressor mites or on clean plants, means that they were not previously infested by any spider mites. And what we see is that the suppression of plant defenses improves the performance of suppressor herbivores, but it also improves the performance of the heterospecific competitors. So we can also consider ecological costs. So if the resources available due to suppression can also be used by competitors. And however, these hypotheses have been discussed in literature, this remains to be empirically tested. So we predict if the suppression is physiologically costly, it will be counter-selected in the absence of plant defenses, and if it's ecologically costly, it will be counter-selected in the presence of competitors. And to study this, we used spider mites on tomato plants. So spider mites are world pests, very famous, and they have high fecundity and short generation time, traits that can allow us to follow the real-time evolution of these populations. T-Evans and T. urtica are closely related species that can occur in the same host plants. And for this study, we used T-Evans, the outbred population, which suppressed defenses, and we used T. urtica as a competitor. So tomato plant is just one of the plants that spider mites can use. And similarly to what happens in other plants, the plant defense response is regulated by jasmonic and salicylic acid hormones. So to study our questions, we follow the real-time evolution of suppressor populations in environments where the benefits of the suppression are expected to be lower than the costs. So here we have three selection regimes. As a controlled selection regime, we evolved suppressor population on plants with intact plant defenses. On the second selection regime, we evolved suppressor populations on plants with impaired defenses, which means the environment where the suppression has no benefits. And on the third regime, we evolved suppressor populations on plants with intact defenses, but in the presence of competitors, meaning that the benefits can be shared by the competitor. So after 14 days, we transferred each suppressor population to a new box with the same environment conditions, and we repeated this for 26 generations. In each transfer, the competitor species was added from the base population, so that's ensuring there is not evolving in the presence of suppressor populations. So just to summarize, we have a control selection regime. The second selection regime allows us to detect physiological costs, and the third selection regime allows us to detect the ecological costs. And we performed the experiment at three different generations, but the mites were not reared in the common garden prior to testing. Okay. So we performed two types of experiments. In one experiment, we measured the suppression levels, and in the second experiment, we measured the fecundity of evolved population to see if the potential costs are expressed on this trait. So for the first experiment, we measured the suppression level by measuring the effect of priming by evolved populations on fecundity of an inbred line. So here is the setup of these experiments. We basically divided a leaflet in two parts using a lanolin and a glue. And so we infested the bottom section with 25 ppm of each selection regime. And 2 days after inducing a response, a plant response, we infected the top section with 5 females of inbred line to read the effect induced by the evolved populations. And to estimate this, we counted the eggs laid by the T. evansi inbred line 2 days after. So, before showing you the results, I just want to explain what kind of results we can expect. So, on the y-axis, we have the number of eggs laid by females of the inbred line, And here we have the control treatment consisting of uninfested plants, which means that only the top section was infested with the 5 females of the inbred line. And we compare the populations to this control. If the number of eggs are above the control, it means there's suppression. If it's below, it means there is no suppression. So here we have the results of the control selection regimes, and we see that the plants that were primed by mites from the control selection regimes, the females from the inbred line had laid more eggs than the uninfested plants, which means that the control selection regime maintained the ability to suppress plant defenses. But when we look at the mites that evolved with impaired defenses, we compare it to the control selection regime, and we see that the females from the inbred line decreased the number of eggs. Okay, so it means that evolving on plants with impaired defenses resulted in decrease of to suppress plant defenses, which confirms the presence of physiological costs. And we see similar results for selection regime where mice evolved in the presence of competitor. So there is a significant decrease, which means that there are ecological costs. Concerning the second experiment, so we measured the fecundity of evolved populations on plants with impaired defenses to detect if the costs are expressed in this trait. And what we expect for this experiment is that the mites that evolved a decreased ability to suppress will perform better than the mites from the control selection regime. So we basically cut small leaf discs. We put one female of evolved population for 2 days, and then we counted the number of eggs. And when we look at the results, surprisingly, we don't see any significant differences between the selection regimes, which means that the physiological and ecological costs that we detected are not expressed on fecundity, but maybe are affecting other traits that we didn't measure. But we also measured the fecundity on plants with intact defenses to see the response in the ancestral environment. And what we expect in this case is that plants that decrease the ability to suppress will perform worse than the control selection regime, where mice, as I said previously, still suppress plant defenses. Interestingly, here we see that only evolving with competitors resulted in a reduced fecundity. And this result suggests that the distant mechanisms underlie the costs that we detected in our experiment. So just to summarize, to our knowledge, this is the first study showing that plant defense suppression entails both physiological and ecological costs. And such a fast response indicates there is a variability of suppression in the ancestral population, and that the physiological and ecological costs are based on distant mechanisms. So to summarize, our results lend support to the role that costs play in the variation of strategies observed in herbivores to cope with plant defenses. And so just to finalize, I just want to thank the MySquad team for the amazing help in my daily work and to perform all the experiments, experimental evolution. And thank you all for listening.
Thank you, Mariya. Questions for Mariya?
I may have missed it, but did you look at plasticity at all? Plasticity of the response?
So, I don't know if I understand your question, but
So you, in the summary, you were saying you thought there was a lot of variability in the ancestral population, but could there also be a plastic response as well?
Yes, so as we didn't perform common garden in our experiment, so basically what we see here is a plastic response, meaning that if we transport mites if they move to another environment, they can recover their ability to suppress, so yeah.
Any other questions? Great.
Sorry, I can hear you very well.
What did you enjoy the most?
Well, well, first I enjoyed working with my team. And so I also like the experimental evolution methodology, so for me it's very interesting, and I like to work with spider mites, I don't know. So, yeah. Thank you.
Hi everyone, I'm Emily Longman. I'm a postdoc at the University of Vermont, and I'm really excited to talk to you today a little bit about some of my dissertation work at the University of California, Davis. And I just have to apologize, I've kind of lost my voice over all the talking over the last couple days. So if you can't hear me or couldn't understand something, please come up and talk to me afterwards. I'm happy to talk more about this work. The field of eco-evolutionary dynamics analyzes the reciprocal impacts that ecological and evolutionary processes can have on one another on contemporary timescales. And although this emerging field is getting a lot of attention, I think there are still a lot of gaps in our knowledge. I think the biggest unanswered question is if these feedbacks are important or trivial in nature. Most experiments have been performed so far in either lab settings or in mesocosms. And I think these experiments are a really important first step for studying eco-evolutionary dynamics. However, field experiments are needed to determine whether eco-evolutionary feedbacks are swamped or amplified by external factors in more complex natural environments. Second, many of the eco-evolutionary dynamic studies so far have generally focused on interactions between pairs of species and have seldom tested for their effects at the community or ecosystem level. Also, most experimental studies of eco-evolutionary dynamics, including studies on many of the taxa that we've heard a lot about over the last several days, have generally actually compared two or more locally adapted populations or divergent ecotypes. This approach relies on substantial phenotypic variation that has evolved through divergent selection imposed on separate populations over decades to a century or more. In contrast, there have been increasing calls for studying the dynamics of systems where there's no separation in time between the evolutionary and ecological processes under consideration. One promising approach for studying eco-evolutionary dynamics is to explore whether abiotic and biotic variation acting over rapid timescales from months to years might impose selection on existing within population variation and functional traits, specifically traits that have links to community dynamics. And I think the Nucella canaliculata system seems to be an ideal system for testing for the community level consequences of eco-evolutionary dynamics. These intertidal snails prey on a variety of barnacle and mussel species. In addition, previous research has shown that this predator exhibits substantial variation in foraging traits and that drilling ability has a heritable basis. A common garden experiment has shown that snails from California, which are on the left-hand side of the red line, readily drill mid-sized Middleus californianus mussels, while those from Oregon, which are on the right-hand side of the red line, generally cannot. But what I really want to highlight is this Bodega Marine Reserve, or BMR population, which contains a mix of drilling phenotypes that seems to vary both among and within Nucella canaliculata families. These snails have a relatively fast generation time and have really high early life mortality. Upon hatching, these juvenile snails are really small and are dependent on a supply of newly recruited prey that are spatially and temporally variable in their abundance. In addition, Bodega Marine Reserve is located along a region of the coast that is known to have high interannual variation in recruitment of both barnacles and mussel species, as well as have high variation in upwelling with high variability in pH between years, which could result in temporal variation in mussel shell thickness. Due to the substantial environmental variation at Bodega Marine Reserve and the high early life mortality in Nucella, there's a potential for strong selection to occur within each generation. In addition, previous work has shown that Nucella can alter things like commuting processes, including mussel bed succession. So I was interested in testing if eco-evolutionary dynamics were present in this Nucella system. I hypothesized that temporal variation in prey could impose strong selection on within population variation in drilling phenotypes, and subsequently that variation in drilling phenotypes could impact mussel bed succession. To test this hypothesis, I conducted a selection experiment where I reared newly hatched dog whelks or snails on different prey treatments, mimicking a situation in nature where cohorts of snails would hatch out and encounter different prey communities. So snails were hatched in the lab, and I split snails from each egg capsule cluster or family of snails among 4 different diet treatments for a 3-month-long selection phase. The diet treatments were a controlled diet of very thin-shelled Middleist trocellus mussels, shown in yellow. I had 2 treatments of Middleist californianus, coming from different regions along the coast. And these treatments were used to represent differences in shell thickness, since previous research had shown that mussels, adult mussels from Bodega Marine Reserve, or BMR, were thicker than those from Soberones Point, SBR. And then my last treatment was a treatment of acorn barnacles. So after 3 months on these diet treatments, I assessed mortality for each family. The mortality of the snails varied among the diet treatments, with snails based on Middleist trocellus, shown in yellow, having the highest survival, and the snails based on the thick-shelled Bodega Marine Reserve Middleist californianus, shown in red, having the greatest mortality. After the 3-month selection phase, I then reared all of the snails out on a common diet of Middleist trocellus until they were adults, at which point in time I ran a laboratory scoring experiment to quantify the snail's drilling capacity in the lab. So I tested individual adult Nucella canaliculata on their ability to drill mid-sized Middleist californianus over the course of a 100-day experiment. What I found is that early life diet had substantial effects on the percentage of surviving Bodega Marine Reserve snails that were strong drillers. Drilling success was highest for those snails that were raised on the Bodega Marine Reserve Middleist californianus, shown in red, and as a reminder, these were the snails that had the highest early life mortality. To test for the ecological consequences of these divergent phenotypes, I did a field outplant experiment where I assessed whether variation in drilling phenotypes would have ecological consequences on mussel bed succession. I predicted that dog whelks would slow succession compared to reference plots where dog whelks or snails would be excluded. And second, that dog whelks and snails with a greater proportion of strong drilling phenotypes, which were shown to be those snails based on the Bodega Marine Reserve Middleist californianus in our lab experiment, would drill more in larger mussels and would ultimately slow the progression of succession. So I cleared mid-intertidal sections at Bodega Marine Reserve. I then let these communities develop naturally for a year, at which point in time I installed cages with removable lids into these regions. So the cages were in a block design where I had 5 cages placed within a given region, where each block represented snails from the same family, with each cage randomly being assigned to contain snails from one of the 4 diet treatments, plus there was 1 cage that was a reference cage that did not get any snails. So if I take off the lid on one of these cages, you can see that the plots at the start of the experiment contain a mid-successional community containing a mix of acorn barnacles, gooseneck barnacles, and a few small mussels. I chose this stage to start my experiment because I thought this would be the point in time that these snails could have the largest impact on the trajectory of succession. So at this point in time, I then placed 5 lab-reared snails into these cages. And then every 8 weeks for the next year, I would go out, remove the lids, and photograph the plots to assess percent cover of sessile species. So what's being shown here is the progression of just one cage through time. So I visualized NMDS graphs for each experimental check, with this figure being the community composition right before I outplanted the snails. So each dot represents the community composition present within a given cage, and the color represents the treatment, with gray being the reference cages and the 4 brighter colors representing the early life diet treatments of the snails placed within those cages. So ultimately, this ordination plot illustrates that the community composition did not differ in the experimental cages at the start of this experiment. So these are the ordination plots through time, going from the top left to the bottom right. And you can see that over time, all of the dots and ellipses move towards the region of the NMDS graphs that represent muscles in the top right corner, ultimately highlighting the progression of succession over time. The trajectory of succession ultimately diverged most clearly for the reference cages with no snails, which again were the dots and ellipses in gray. But the big question was how the colored ellipses differ. And although there were some differences on community composition among the treatment groups, there was substantial overlap and no clear differences in the trajectories of succession. So despite finding differential mortality among the diet treatments, suggesting the potential for selection, and the fact that we saw differences in adult drilling capacity in the lab, eco-evolutionary feedbacks were relatively weak when tested in a natural field setting. And I don't think this is an artifact of just our system. I think there were a number of factors and constraints that may have driven this finding, and I think these factors likely apply more broadly to other systems. I think eco-evolutionary feedbacks are likely often swamped in the field by the overriding effects of substantial spatial variation in environmental conditions. So on rocky shores, recruitment and wave exposure often vary substantially over relatively fine spatial scales and may affect the rates of succession. Against this backdrop of substantial variation in environmental drivers, the influence of intra-population variation in drilling traits on the rates of succession may be comparatively minor. Second, given that our focus for this project was having no separation in time between the ecological and evolutionary processes, we focused on the variation present within one population. However, there was likely a limited range of within-population variation, especially when compared to that being found across populations in this species. Thus far, many experiments that test eco-evolutionary dynamics have compared the effects of locally adapted populations or divergent phenotypes. A similar approach has been taken in the field of plant community genetics, where plants are collected from distant diverged populations, and their effects on insect communities are quantified in a common garden experiment. These approaches often create a mismatch in the spatial scales used to test the relative importance of environmental versus genetic influences on community processes. Lastly, eco-evolutionary feedbacks might be weaker than expected in nature if selection on a population is not strong enough to produce extreme divergence in functional traits. The existence of wild populations comprised of phenotypically diverse individuals suggests that past selection has not been strong enough to eliminate variability in these traits under more realistic selection regimes. Thus, even when there's substantial selection, like the selection that I performed in this experiment, there were likely some strong drillers present in all of the diet treatments. And the maintenance of this variability likely dampens eco-evolutionary effects in this community. So despite finding evidence that early life diet treatments generated divergence in drilling traits, the effects on these community dynamics in the field were relatively weak. And we think this likely applies much more broadly, where a range of factors and constraints likely dampen the strength of eco-revolutionary dynamics in natural communities. I'd like to thank all the people who helped in the field and lab for this project and my funding resources. Questions?
Hi, I'm Amina Mohamed. I'm a second-year PhD student with Andreas Lopez Sepulcre and Swan Gordon at Cornell University. Thank you so much, Emily, for explaining eco-evo dynamics so beautifully, but I might disappoint you with the common guppy, common garden experiment. So I'm very excited to talk to you all about some very new results from an experiment looking at how guppies manage nutrients and what effects they can have on the ecosystem. So consumers, when they eat food and grow and accumulate body mass, they act as sinks of nutrients, and when they excrete and adjust and ultimately die, act as sources of nutrients, which can then be uptaken by primary producers, and thus they play a very important role in nutrient recycling in the ecosystem. And a great way to look at, is it too loud? I don't know, okay, maybe, okay. A great way to look at nutrient dynamics and flow of nutrients through ecosystem is to just focus on the elements. We know that elements, they follow mass balance principles, and so it's easier to account what goes in and what goes out. And ecological stoichiometry is essentially the framework that uses mass balance principles to look at ecological interactions. Often, the environment that an organism is in might not be the ideal for that organism. So they try to actively maintain their internal nutrient composition so that they survive. And stoichiometric homeostasis refers to this process. The degree to which an organism maintains their elemental composition might vary. And you can look at this by looking at the relationship between resource stoichiometry and body stoichiometry. So if the body stoichiometry is exactly same as what you eat, then there is no homeostasis, and at that point you're probably not even alive. But if you change your body stoichiometry according to what you eat a little bit, but within some range, we can say you show flexible homeostasis. Whereas if your body stoichiometry stays almost the same, regardless of what you eat, you can say they show strict homeostasis. So generally, autotrophs are considered to be flexibly homeostatic and heterotrophs are considered to be strictly homeostatic. But studies have shown that even within heterotrophs, the degree of homeostasis varies. So you can see that different slopes indicate different degree of homeostasis. So what might be the factors that is selecting for strict versus flexible homeostasis? So this is like one hypothesis. It might be the nutrient concentration in the environment that is affecting this. So if the nutrient is more limiting in the environment, you might have to actively try hard by excreting less of that nutrient or assimilating more of that nutrient to maintain your ideal elemental composition, and you might show strict homeostasis then. Whereas if the nutrient is more not limiting, it's more abundant, you probably don't have to try hard, and you can probably just show flexible homeostasis. There are other factors that can also affect the degree of homeostasis, like growth rate. If you are growing faster, you need more phosphorus. That might change how your body stoichiometry varies. And if you are storing more of some nutrient, for example, in your bones or something, that's called luxury uptake, that might also influence your degree of homeostasis. So the question I'm interested in answering was, how does diet quality affect stoichiometric traits in guppies from low and high predation environments? I know I haven't introduced my model system, which probably doesn't need much introduction, but Trinidadian guppies or Porcelia reticulata is a great model system to answer questions on evolutionary biology. Studies have shown that they rapidly evolve life history traits when translocated from one environment to another. So in the wild, in their native habitat, they are seen in two distinct environments, and environments when they coexist with predators who eat them, they occur at a lower density, and there is more resources around, there is less competition for resources, and they have access to good quality food, and they usually eat nutrient-rich food like invertebrates. Whereas when they somehow reach upstream, they don't coexist with predators, they occur at a higher density, and there is more competition for resources, and then they have to eat whatever they get, and they often have to eat crappy food like algae or detritus, which is nutrient poor. And studies have also shown that low predation guppies, I might use LP, HP for low predation, high predation guppies. So studies have also shown that low predation guppies have a higher imbalance between what they eat and their body. So even if they eat bad food, they somehow keep their nutrients ideally. So I had two hypotheses going into this experiment. The first one is that guppy stoichiometric traits have a genetic basis. If this is true, I would expect whatever trends that I saw in the wild to show even in the lab after a couple of generations. And my second hypothesis is that higher competition for resources would select for higher homeostatic ability. If these hypotheses are true, this is what I would expect to see. Triangles would always stand for high predation guppies, and circles stand for low predation guppies. So when I change the food quality, I would expect low predation guppies to show strict homeostasis, and high predation guppies to show more flexible homeostasis. Whatever is not assimilated is excreted out, so I would see a complementary pattern. I would expect to see a complementary pattern in the excretion. So to test these hypotheses, I used guppies from one of the streams, adipose streams, and I used F3 generation females from high predation and low predation environment, and reared them under high or low quality food. So what I mean by high or low quality food is when I went to Trinidad, I collected high quality invertebrates and low quality algae and detritus from the wild and looked at their carbon, nitrogen, phosphorus ratio, and I tried to mimic that in the lab with artificial diet. And I reared them under this treatment for 12 weeks. During that time, I measured their growth, and at the end of 12 weeks, I measured the ammonium and phosphate excretion, and after that I sacrificed them, dried them, ground up, and did some body stoichiometry analysis. So let's see what I saw. It's very fresh. So initially I'll show body stoichiometry results and then excretion. We'll start with nitrogen. So this was my expectation, and this is what I saw. X-axis is food quality, y-axis is percentage nitrogen content in their body, triangles high predation, circles low predation. So we can see that high predation guppies showed strict homeostasis and low predation showed more flexible homeostasis. So it was against my predictions. And when I actually did more literature search, it was, I saw that actually high predation sites were nitrogen limited, and it was cool to see that high predation guppies showed strict homeostasis. For phosphorus, this was my expectation, and I saw results that supported that. You can see that the low predation guppies are more homeostatic. It has a smaller slope, and when I looked at the literature, low predation sites were actually phosphorus limited, and low predation guppies showed stricter homeostasis. For excretion of nitrogen, this is what I saw. Again, x-axis food quality, y-axis excretion rate. You can see that the triangle or high predation excreted more nutrients than the low predation guppies, and this also supports the trends in the wild. The high predation guppies excrete more nutrients, and this might show that there is some genetic basis to this trait and it could rapidly evolve. For the phosphorus excretion, I didn't really see any significant pattern with food quality or population, and you can also see there's a lot of noise. So I'm doing this replication with another stream, and hopefully with more data we'll see more patterns. So coming back to this initial framework of hypothesis I had made very simple predictions on the basis of competition and resources, but I didn't really look often. I mean, real systems are more complex and there's a lot going on. There might be variations in nutrients according to season, according to sites. Different nutrients might be different, but it was very cool to see that the framework still the results still supported my framework. That when it was limiting nutrients, it showed that strict homeostasis. But there's still so much more to explore here. And for future, I have replicated this with another stream, and I'm at the last stages of it. And I would also like to use modeling techniques to model an optimum nutrient management strategy and how that can have an effect on on the ecosystem nutrient cycling. And I mentioned that growth rate is also something that affects degree of homeostasis. So I would also like to track the changes in stoichiometry through ontogenic stages. And yeah, with that, I would like to thank my wonderful advisors, lab, friends, undergrads, and my funding resources. And yeah, I'll take questions. Yes, Lacey.
Would you expect any of the other aspects of the ecology between the low and high reaches of predation to affect nutrient uptake, like shoaling behavior, kind of these personality behavioral traits that could change, luxury uptake, that kind of thing?
Yeah, I'm sure there are a lot of factors that would affect the resource availability and also uptake. But that's why I decided to go very simple and do lab experiments. But like Emily also said, the real life is more complex, and we don't even know how strong these eco-evo feedbacks are. But that's a great question, Lacey. Thanks.
There's differences in the stoichiometric ratio. Is that because they differ in their relative mass of tissue versus bone and all those kind of, like, is that what we're thinking? Sorry, I don't know even how to
No,
pronounce that.
that's a great question. All my results are corrected for just body mass because all these stoichiometric traits vary allometrically, but I haven't really looked at different proportions of different organs or anything, but there should be something, yeah, going on. That's a great question, thanks. Yes?
Did you measure ejection?
I did not. So excretion as a result of metabolic waste and ammonium and phosphate can be directly uptaken by primary producers, and it's much easier to look at excretion. I just have to look at the concentration in the water, but egestion for tiny guppies, it's a hard thing, but that would also explain things that I'm missing. Great question. Thanks. Yes, Max.
Would you expect to see the same patterns in males?
I don't know. So I ignored males and just used females because females get bigger and like they have a higher effect on the ecosystem. But males are kind of weird. I don't know. Yeah, good question. Thanks. Yes.
Are there some differences between these two populations in terms of like metabolic rates, and did that affect the like stoichiometry somehow?
Yeah, I am completely ignoring some of the elements, and metabolic rates is also a big part I should look at when I talk about degree of homeostasis. And then we also know there is high predation, low predation, that this having a predator would affect their stress, that would affect their metabolism, and that's also an important thing to look at. But since I am controlling for all of that, not really, don't know what's happening, but yeah, great point. Thank you so much. Yeah.